Hello and hi everyone. I hope you are ready to learn my topic on transgenic animals. Okay, let's begin. Do you know that the first successful transgenic animal was a mouse? Okay. What is a transgenic animal actually? You know that DNA contains many genes that are stored in the cell in a structure called the nucleus. The genes store information that regulates how bodies form and function. The genes can be altered artificially, so some characteristics of an animal are changed. It is called gene editing. Transgenic animal is one that carries a foreign gene that has been deliberately inserted into its genome. Such introduced genes are called transgenes. The foreign gene is constructed using recombinant DNA methodology, inserted into the germline of the animal, and it can be transmitted to the progeny through mating. Okay, each transgene contains a promoter, which determines in which cell the transgene is active, an intron, a protein coding sequence, and transcriptional stop sequence. Okay, here is an article showing you the example of transgenic mouse creation. In this article, the foreign gene that was added in the mouse is ROC2. Okay, so the researchers in this paper, they wanted to see if addition of ROC2 gene may influence cell morphology or cell proliferation. Therefore, in the future, this gene may be targeted for therapy, maybe in certain diseases such as cancer. So here comes the question, why are transgenic animals being produced? Okay, the transgenic animals can be used as a disease model to study the mechanisms of disease, to find an effective treatment for disease. Example in the cancer research. Alright, second, the transgenic animals can be used for food and agriculture purposes. Right. For example, transgenic cows that can provide more nutrients in their milk or even their meat. Okay? So the real question is how you can produce these transgenic animals. Of course, by construction of a transgene to be inserted into the genome of the animal. And here are the three methods to produce the transgenic animal. First, DNA microinjection, retrovirus-mediated gene transfer, and third, embryonic stem cell-mediated gene transfer. I will explain each method after this. Alright, first, DNA microinjection. The mouse was the first transgenic animal that has been produced by this DNA microinjection method. In this method, it involves a transfer of a desired gene construct. Okay? The gene construct can be a single gene or combination gene from another member of the same species or from different species. Nowadays, we have an available of commercial gene constructs that are made by scientists, and you can purchase chest it for your research. Okay, the gene construct then will be transferred into a pronucleus of a reproductive cell. For the flow method, first the egg cells or reproductive cells are collected. Then the transgene is injected into the pronucleus of the egg cell. The eggs that have been injected with transgene are kept overnight in an incubator to develop a two-cell stage. Then the eggs are implanted into the uterus of a pseudo-pregnant female animal. Okay, you may watch this link about DNA microinjection method for further understanding. I will share the link to you in the comment section below, or you can find it in the UKM folio under this topic. Alright, you might wonder why we need to implant and let the cells grow in a pseudo-pregnant animal, right? Okay, I put mouse as an example here. First, if you remember, we cannot use the same mouse that has donated the egg cells or the reproductive cells because the mouse has been sacrificed in order to collect the egg cells. Hence, we need a surrogate female mouse, but before you can impregnate the surrogate mouse, you need to mate the female mouse with a male mouse, which had undergone vasectomy. Okay? This is to make the mouse pseudo-pregnant. Okay? Because they are mating with the male mouse that has undergone vasectomy. So this process is important to make the female mouse is ready, not only their physical but also hormonal changes are also ready to have a successful progeny of transgenic mouse or transgenic animals. Okay? Second method is retrovirus-mediated gene transfer. You know that most viruses have either RNA or DNA. Retrovirus is a virus that carries its genetic material in the form of RNA rather than DNA. First, you can insert the desired transgene into the retrovirus genome, which acts as a vehicle. The RNA is transcribed through the virus' own reverse transcriptase into DNA in the infected cells. The infected cells will be cultured, and those expressing the target gene will be inserted into the target cells, such as embryo cells of an animal to produce the next transgenic progeny. Okay? Alright. I will share the video on the retrovirus-mediated gene transfer. The link also can be found in the UKM folio. Alright, the third method is the embryonic stem cell mediated gene transfer. In this method, embryonic stem cells are obtained from the inner cell mass of a blastocyst. Then you can insert the construct transgene into the embryonic stem cells. The transgenic stem cells are grown in vitro via cell culture, then implanted into a host uterus We let it grow and produce a transgenic progeny. Okay? Here is a flow image showing you the step-by-step method in embryonic stem cell mediated gene transfer. Okay? Alright, for your activity, I want you to search a research article that has done an experiment by using a transgenic animal. And then, from the research article, you read and please state what is the transgenic animal that the paper used. Okay? Please write your answer in activity session under this topic in UKM folio. Okay? Example answer here, like if the paper is by Mastery et al. 2020, and then you state what is the animal. Such as transgenic mouse here. If it is transgenic fish, put transgenic fish. Okay? Here are some examples of transgenic animals that have been produced, such as transgenic fish, transgenic mouse, and transgenic cattle or cow. Each of these animals serve a specific role, either for food, for nutrients, or as a disease model. Alright, let's talk about gene knockout. I put example animal again, a mouse in here, okay? For transgenic animals, you may either activate your gene of interest or you may also inactivate the gene. You can inactivate the gene of interest by gene knockout. Okay? A knockout mouse is genetic manipulation where we delete a gene or disrupt a gene's function in a mouse model organism. Okay, the question here, why do you need a gene knockout model? Because it is useful to study development, behavior, physiology, and also as a model to study certain human diseases. In order to produce gene knockout mouse, the embryonic stem cell containing a knockout mutation is introduced into the mouse embryo. The mouse progeny will be a chimeric mouse, which contains both the transplanted embryonic stem cell and the host cell. Okay, combination of both. So the chimeric mouse is method to assess whether the mutation is incorporated into the germ line to their progeny. After that, the mouse progeny that is heterozygous for the knockout gene is mated with other heterozygous mouse in order to produce homozygous knockout mouse. Okay? So you will get total knockout gene expression. Okay. Here is the link of video showing you how the knockout mouse is produced. Okay, the method is not much different than the embryonic stem cell mediated gene transfer. It's just that the construct transgene will be different, okay? Where the gene of interest will be silent or knockout for this model. Alright? Okay. Another question comes up. But what if you want to knock out the gene to just a specific cell or at a specific stage of development in the animal? So how are you going to do it? Okay, as most genes function in different parts of the organism and at different times, so a knockout mouse may die or have defects in various tissues prior to the stage to be analyzed. Okay, so then how? Okay, scientists created a clever technique using a method called CRISPR model. Okay, in this CRISPR model, log P sites are the site-specific DNA recombination, and the enzyme is called CRISPR, which catalyze the recombination between them. Okay. So here are some explanation on how the CRISPR work. Okay. The CRISPR recombinase is expressed under the control of a promoter that is active in a desired cell type. Then a gene of interest is blocked or flanked with log P site. Okay, if you look at the right image here, okay, to produce or to do this CRISPR model, you need two types of mouse. First, a mouse with the CRISPR system, and second mouse with the log P system. The mouse with the log P system has a gene of interest, okay, that flank, okay, with the log P site, okay. There is a target gene that you need to knock out, right? So you mating these two mouse, CRISPR and log mouse together, and you can produce a CRISPR log mouse, alright? So after you produce the CRISPR log mouse, then the progeny can either be a knockout gene where the CRISPR recombinase is activated and the target gene is being silenced or knockout, or it can be another progeny of mouse where the self-selecting of active CRISPR recombinase where the target gene is untouched. Right? So in order to confirm which progeny has successfully knocked out gene, you need to do the PCR to see the expression. Okay? So basically in CRISPR system, the CRISPR recombinase enzyme binds to the log P site and it will cut the DNA in between them and knocking out the gene of interest in the desired cell type. Okay, because the CRISPR already has a promoter that is targeting to the desired cell type. Okay, let's say skin, so the promoter is on the skin, right? Okay. Currently, we have new technology in genome editing that is able to produce transgenic animals. The emergence of site-specific nucleases has opened new windows for rapid development in transgenic animal models. There are zinc finger nucleases, transcription-activated light receptor nucleases or TALEN, and the newest one is CRISPR-Cas9. Alright, among these three, CRISPR-Cas9 is far more superior, as CRISPR-Cas9 is not only easy to perform, it is also efficient and multiplexable. It can also be applied for gene knockout and gene knocking. Moreover, this CRISPR-Cas9 can be used in animals, in plants, and some studies even claim that they can use this technology to humans as well. But of course, this issue is still in debate until now. Alright, for CRISPR-Cas9, it consists of an RNA-guided DNA endonucleases, which is Cas9, and corresponding guide RNA, which is CRISPR. So the question is, how CRISPR-Cas9 lets you edit the DNA? Okay, as I mentioned earlier, Cas9 is the enzyme that cuts the DNA, and the guide RNA will direct the Cas9 to the target DNA. Okay? Because the guide RNA, it contains a sequence that will match with the target DNA. So when Cas9 and guide RNA stick together, they will stick out to the target DNA. Alright. And then Cas9 unzips the target DNA and the guide RNA matches up with the target DNA. And if the match is complete, the Cas9 then uses molecular scissors to cut the target DNA. Once it being cut, the DNA can be disabled or altered. Now the scientists can edit the gene how they wish. Either they want to do gene knockout or gene knocking. So basically, CRISPR-Cas9 is like a system where you correct a typo error, okay, like gene editing to gene editing. Okay, as simple as that. Alright. Still on the CRISPR-Cas9 topic, if you watch a movie called Rampage, okay, where the hero is Dwayne Johnson, you can see they talk about the CRISPR-Cas9 technology where they produce a super strong animal in the movie. Okay, but of course, from research papers, the successful animals that have been produced so far from CRISPR-Cas9 technology are only mouse and monkey. Okay, and further research are still ongoing. And you know that in this year 2020 Nobel Prize winner in the chemistry is a founder of the CRISPR-Cas9 technology. Okay? I will share the link of the talk by Jennifer Doudna, one of the Nobel Prize winners, on the CRISPR-Cas9. Okay? So how transgenic animals can contribute? First, in agriculture applications, okay, in breeding, okay, transgenic animals can exhibit desired traits, right, such as increased milk production or high growth rate. Okay. Because traditional breeding is usually time-consuming. So, if we can produce transgenic animals that can have a desired trait, so it can also help in the breeding process, which can be even shorter time and with more precision. Okay, it will help the farmer an easy way to increase their yield. Second, quality. Okay, the transgenic cows can produce more milk, more meat, and even more wool for transgenic sheep. And also for disease resistance. Okay? We can produce a transgenic cow that resistant to mad cow disease. Second, for medical application. Okay? For example, in xenotransplantation, okay, to provide the transplant organ. And then another example as nutritional supplements and in pharmaceutical industry and also in human gene therapy. Okay? And third, for industrial applications, we can produce a toxicity sensitive transgenic animals for chemical safety testing. And there was a study by two scientists from Canada where they combined the gene from a spider into the cells of lactating goats. So they produce a transgenic goat where not only producing milk but they produce a thread, okay, a tough flexible material that can be further used in various applications such as military uniforms, medical, microstructures, and tennis racket strings. However, of course, there are several issues of concern from the development of transgenic animals. One area of concern is the moral status of the animals, animal rights, and animal welfare. Another area of concern is the boundary constructed between what is considered natural and unnatural. Okay, because if these things becomes a norm in our society, then it's hard to differentiate between the natural one and the unnatural one. Also, concerns have been expressed about the risk from long-term impact for human and for the environment. And a major concern is the number of excess animals required to establish a transgenic line and to produce the required number of animals with the correct gene complement. Because when you produce the transgenic animals either by the DNA microinjection or by CRISPR-Cas9, do you think you can produce it by one-time experiment? Okay, it's just for the people that have a magic finger. But you need to try a lot, okay? And use the number of animals, okay, to produce the correct gene that you want. Either you want to knock out or knock in the gene. Here is one of the issue of concern. Alright, if you know, the brand called Michael Kors, Gucci, and other familiar names in the industry, they're using fur from monster foxes. This means that they produce a transgenic fox who are bred to be 5 times bigger than normal foxes. And this is how the transgenic fox looks like. Okay? Okay, another thing is the ethical concerns. Ethical concerns must be addressed as the technology of producing transgenic animals is growing. Thoughtful ethical decision making cannot be ignored by the biotechnology industry, scientists, policymakers, and the public. That's why you need to apply for the ethical and animal ethic application before you can start doing a research on transgenic animals. Okay, that's all from me. Thank you everyone. But before that, don't forget to fill in the attendance form that I share the link in the KM for you. Okay, and which one of these image is natural and which one is not natural? Okay, that's all. Bye.